Did you know that a review commissioned by the United Kingdom government argued that by 2050, 10 million people per year will die due to antimicrobial resistance? Despite antibiotics being mainly used to treat diseases related to bacteria, they have been used also for molecular biology procedures, based on confirmed microorganisms' ability to resist the presence of antibiotics, in order to investigate and produce protein or metabolites. However, this has raised multiple concerns due to the risk it presents towards the environment and human health, since the main threat is the development of an antibiotic resistance among pathogenic microorganisms, especially in case these were contagious among humans. TecCEM team's proposal is to reduce the use of antibiotics in experimental procedures by using isogenol instead when performing genetic modifications. But how exactly are antibiotics used and why isogenol could replace them in experiments? Well, naturally, all microorganisms die when they're exposed to antibiotics, but when we genetically modify them to produce a product of interest, an antibiotic resistant gene is also inserted. Thus, genetically modified microorganisms can grow in antibiotics while others die. Isogenol, the isomer of eugenol, is a natural compound responsible of the antimicrobial properties found in the essential oil of cinnamon and clove. Because of the inhibitory effect of isogenol, a normal microorganism would die, but if we genetically modify them to produce isogenol monoxygenase, they could transform into vanillin and live.
But how was this carried out? First, a proposal was made focusing on finding out which specific compounds were responsible for the inhibitory effect of cinnamon essential oil, particularly on E. coli, and if there was a reported degradation pathway for these compounds, and the final product of this degradation was not hazardous for the bacteria. After analyzing these factors, it was determined that the better option was eugenol, one of the inhibitory compounds of cinnamon essential oil. However, its degradation pathway was complicated since it involved the expression of 5 different genes, so further investigation was needed. Yamada, Okada, Yoshida, and Nagasawa reported in 2008 that the isomer of eugenol, isogenol, could be transformed into vanillin by the action of a single enzyme, isogenol monoxygenase, originary from Pseudomonas bacteria. Therefore, the problem with the degradation of eugenol had been solved. Isogenol antimicrobial activity covers a wide range of gram-positive and gram-negative bacteria, including Escherichia coli, Bacillus licheniformis, Micrococcus luteus, Pseudomonas aeruginosa, Salmonella type B, Staphylococcus aureus, as well as other microorganisms like Saccharomyces cerevisiae and Aspergillus niger, which means that it could lead to the development of a selection marker useful not only for bacteria but also fungi and yeast. Its action mechanism consists of altering the integrity of inner membrane of bacteria, causing spillage of fluids and cell death.
This alternative represents advantages in comparison to antibiotics because isogenol's low cost, it is not hazardous to the human health nor to the environment, and it possesses a high temperature resistance that allows medium sterilization with it. Whereas antibiotics are expensive and are sensitive to temperature. Nevertheless, there are reports of some microorganisms that are able to metabolize it, such as Pseudomonas. They use an enzyme called isogenol monoxygenase that transforms it into vanillin. Isogenol monoxygenase from Pseudomonas putida reported by Yamada et al. In 2008. It doesn't show that it has another compound that can be inhibitory for E. coli growth.
For the construction of the enzyme expression system for the isogenol resistance, two genetic constructs were designed, one inducible by isogenol and the other regulated by a constitutive promoter. This in order to test the efficiency of both systems for the expression of the enzyme.
Both coding sequences have a signal peptide, POB, to directly isogenol monoxygenase to the bacteria periplasm. PP22, the inducible construction, is 2,807 base pairs long, includes a bidirectional promoter that regulates the transcription of iso, the coding gene of isogenol monoxygenase, and IMR, a transcriptional activator reported by Ryu, Seo, Park, Chung, Sadowski, and Her in 2012. And lastly, two different terminators were added, one for each coding gene, the terminator of the tryptophan operon and terminator RRNBT1. NC on its own is 1,712 base pairs long. It is a shorter sequence that shares the iso gene and the RRNBT1 terminator in common with PP22, the longer sequence. However, this construct includes the strongest constitutive promoter reported, the BioBrick G23100, as well as a ribosome binding site.
As a first stage of the project, experimental procedures were designed focused on ensuring that isogenol was adequately homogenized in the medium, considering that this is an allyl compound. Since it was noticed that isogenol was not dissolving in the mediums despite the small amounts that were being used, we decided to try to emulsify it with a polytron homogenizer. Unfortunately, this procedure was not effective. Besides the sterilization protocol used for the polytron, all mediums got contaminated. Therefore, other options needed to be explored. As the mechanical emulsification method was not successful, an alternative twin 80 emulsifier was added to liquid culture mediums. Twin 80 is a non-ionic surfactant that is widely used as an emulsifier in cosmetics, pharmaceuticals, and food products. This by dissolving all its compounds. However, with the Twin 80, no improvement was observed in the homogenization or emulsification of the samples. For this reason, and considering its toxicity for E. coli, it was decided that it was better to not use this compound in the medium formulation. In order to avoid using other compounds that could have an inhibitory effect on bacterial growth, it was decided to go back to mechanical methods. Therefore, emulsification was carried out first by vortexing and second by sonication of the media.
A zeta sizer analysis was performed in order to check out the treatment that could provide a better homogeneity of the media using 4.5 millimolar concentration. This particle size variation of isogenol in LV media with zeta sizer is recommended to have a particle size lower than 100 nanometers and with few peaks. These results indicate that the quantity of particles have an homogeneous size between all of them. Once good results were obtained concerning the homogeneity of the culture mediums, one question remained. At which point of the procedure was it better to add isogenol into the mediums? Hence, the efficiency of three different treatments was tested. A. Addition of non-sterile isogenol before sterilization. B. Addition of sterile isogenol after sterilization before plating, similarly to antibiotics. And C. Addition of non-sterile isogenol after sterilization before plating, similarly to antibiotics. This table shows the 6 concentrations of isogenols that were tested. After analyzing the growth behavior of serial dilutions to the power of 10-4 and to the power of 10-5 of E. coli DH5 alpha in each treatment, it was determined that results from treatments A and B were very similar, and due to inconsistencies, treatment C was discarded. Despite the similarity between treatments A and B, it was decided to keep up with treatment A since it is not necessary to manipulate the medium after removing it from the sonicator.
Once it was established the method of isogenol incorporation to the medium, an analysis of its inhibitory effect in regard of the minimum inhibitory concentration was developed. 6 different concentrations of isogenol were tested with 2 of the most used Escherichia coli strains, DH5 alpha and BL21. This figure shows the results of E. coli BL21 with no genetic modifications growing in different isogenol concentrations. It is shown that both negative controls had grown bacteria with big thick colonies. With this, the other plates had an increment on the colonies, but with a smaller size and opacity. Along with the increment of isogenol concentrations, in the highest concentrations no growth was observed since the isogenol inhibit the growth of these microorganisms. After analyzing the results, the 4.5 millimolar concentration of isogenol was defined as the minimum bactericidal concentration for solid medium. This experiment was performed both with DH5 alpha strains. Nevertheless, it was repeated several times. Not concise results were obtained.
As both of our coding sequences were synthesized, the inducible biisogenol and the constitutive promoter, the characterization of their behavior started by ligating both of them into pjet 1.2 vector, in spite of this being a cloning vector and not an expression vector. The selection of this vector residing on its ends facilitates ligation and resistance of ampicillin. After this, both vectors were used to transform chemically competent cells DH5 alpha and BL21 E. coli cells. Transformed cells were plated on LB with ampicillin for assuring the growth of them. Then they were incubated at 37 Celsius for 12 hours.
The table shows the results from this transformation. Once the transformed colonies were obtained, a plasmid extraction was performed with a quick plasmid mini-prep kit from Invitrogen. The next step of the characterization was performing a series of digestion for the 10 plasmid samples. It was carried out with restriction enzymes XBA1 and SPE1. The use of the same enzymes for all the samples was possible due to the biobrick design of both genetic sequences. The digestion product was analyzed with an electrophoresis, as it is shown in the image. Samples in lanes 7, 8, and 9 were degraded. Nevertheless, the samples in the lanes 4, 5, 6, 10, and 11 show the correct sizes of the digestion. Polymerase chain reaction was executed in order to observe the amplifications of PP22 and NC22 using PFHT22 and PRHT22 primers, and be able to confirm that their presence in the plasmids extracted from our transformed cells, as it can be seen in the figure. Lanes 2, 3, 4, 5, and 6 correspond to PP22. Amplification products can be observed in all of them. Lanes 7, 8, 9, and 10 correspond to NC22. Only lanes 8 and 10 show amplification products. After verifying the presence of the PP22 and NC22 in the transformed cells, the following samples were selected: E. coli DH5 alpha transformed with PP22 and E. coli BL21 transformed with NC22. And with these colonies, the expression of the isomo was evaluated with a complete protein profile by culturing them in LB medium at different concentrations of isogenol. Unfortunately, no difference in the band pattern was observed in either DH5 alpha and DH5 alpha transformed with PP21, nor BL21 and BL21 transformed with NC22, indicating that there is no expression of the enzyme by the technique used. Because of the fact that isomo has an approximate size of 54 kilodaltons, thus a band of this size should be observed in all samples. Additionally, in samples corresponding to transformed cells with PP22 should be also showing a band of about 35 kilodaltons, that is the transcriptional activator, or ISO.
Some advances were made in order to use isogenol as a selection marker, including the establishment of the most efficient protocol for its incorporation to the mediums, which, as the synthesizer analysis showed, consisted of adding the isogenol to the medium, then putting it to vortex for 5 minutes, sonicating for an hour, sterilizing in the autoclave, and finally sonicating for another hour. And also, the minimum inhibitory concentration, or MIC, was determined to be for liquid media of 1 millimolar and for solid media 4.5 millimolar, both having an efficient inhibitory effect on the bacterial growth. Therefore, these concentrations can be utilized when wanting to grow only transformed bacteria with the isogenol resistance. And by analyzing the results obtained, we were able to determine what measures need to be taken in the future in order for this to work. And we established for further experimentation, number 1, to test the coding sequence of isogenol monoxygenase by inserting it in an expression vector, and number 2, to use other isogenol monoxygenase sequences that could be more genetically similar to E. coli and therefore easier for its expression and for the transformation of isogenol to vanillin. By taking into account all of the previously reviewed, we consider this to be an innovative proposal for the substitution of antibiotics in synthetic biology experimental procedures. It could help not only other teams but also other scientists around the world to make big contributions to the progress of science in a more secure way, as well as to develop more integral projects that could do introspection about antibiotics, their use, and the importance of relating them to dual use and biosafety and biosecurity, given that nowadays the increasing resistance of bacteria to traditional antibiotics endangers public health, showing that it is necessary to reduce, if not to get rid of, their use in laboratory practices.